Hello, my fabulous friends. Today we're going to be talking about genetics part 1, the intro to genetics, and how to set up a monohybrid cross. Well, genetics is the study of heredity, how things are passed down from parent to offspring, or parent to child. How does that work? Well, the first guy to actually start taking a look at this, his name is Gregor Mendel. He was an Australian monk, and he lived in a monastery. Now, his days were quite slow. They did a lot of meditation. They did a lot of gardening. They read books. They helped the poor. But in his spare time, he did mostly gardening. That was his passion, was gardening and mathematics. So what he did is he combined them. He looked at peas, and he says, okay, I have 365 peas over here. And these type of peas over here, I have 247 of them. How did I get that many of those, and how many did I get that many of those? And what happens if I mix the two? So, Gregor Mendel is known as the father of genetics. He was the first to predict how traits would be transferred from one generation to another. And he looked at pea plants. Now, he actually did the fertilization. He did two types. He self-fertilized, which he took the pollen from the flower and actually put it on the stamen on the same flower. Now, this would guarantee the same traits crossing with the same traits. But he also did cross-pollination, which he took pollen from one flower and put it on the stamen of the other. Now, this means that this flower could be tall, and this flower could be short. So he could find out what would happen if he mixed those two. Let's get some definitions down first. A character is a physical trait that is inherited. A trait is one of several forms of a character, such as brown eyes is only one type of eye color. A hybrid is when you take two traits and you mix them together, such as you have a tall and a short and you mix it. It's going to be a hybrid, a combo of both. So a monohybrid cross. Mono means one, so we're looking at one character. Now this character could have multiple different traits. Most of the time we look at the ones that have two traits. It's either going to have a dominant trait or a recessive trait. A dominant one that is always going to be there, or a recessive one that's sometimes there. We show the difference by the capitalization or the lowercase of the letters. So looking at one single character, such as height, we show the two different traits by a big T and a little t. Big T is dominant, capitalization is dominant, lowercase is recessive. And we look at three generations typically. We start off with the parent generation, and we cross them to make the first generation, or the F1 generation. Then we cross those to make the F2 generation. Now, P is for parent, and F means filial, which is going to be son or daughter. Now, he looked at all sorts of different characters. He looked at seed shape, seed size, seed color, flower color, pod shape, pod color, size of flower, where the flowers were, all sorts of different ones. So he didn't just look at one thing. And he took his first generation. He decided that he was going to take a tall plant that had been created from tall parents and a short plant which had been created from short parents, and he decided to mix the two. So he cross-pollinated these two plants. Now, when the seeds were produced, he planted those seeds, and he found that every single seed produced by this combination were tall. Well, wait a second. We have a short parent. So where did the short go? We came up with two conclusions. One, factors are passed down from one generation to the next, determine inheritance. We call these traits, or these factors, genes. The two different forms of the genes are known as alleles. And then he figured out the principle of dominance. States that some alleles are dominant and some are recessive. For a dominant, you only need one dominant for it to show. Doesn't matter what this other one is. As long as there's one dominant, it will be dominant. To actually show a recessive, you actually have to have two recessives. So an organism with a recessive trait will only show up when a dominant allele is not present. So when there's two recessives. So what he found was that round was dominant to wrinkles. That round, if there was a round, it would be round. And the only way to get a wrinkle was if there was two little wrinkles. And he found that yellow was dominant to green, and inflated to restricted, and etc., etc., etc. Because he looked at each of these characters. Now, he figured out that there was something called the law of segregation. During fertilization, whether we're talking about people, whether we're talking about plants, whether we're talking about animals in general, or whatever, during fertilization, the male gametes and the female gametes randomly pair up. So over here, we have Dad, and he could either give a big A or a little A. Well, he creates two types of sperm. He has a sperm that has a big A in it, and he has a sperm that has a little A in it. Well, Mom can also either give a big A or a little A. And one of her eggs will meet with one of his sperm. So let's look at this guy. Let's say this big A from Mom comes over and meets with the big A from Dad. Well, that will create an offspring that has two big A's. But this big A from Mom could also randomly meet up with this sperm that has the little a to create a hybrid, a big A and a little a. So there are three combinations that we can come up with. Big with big, big with little, or little with little. Now crosses and Punnett squares. To record results for crosses, we see the dominant is always written first. If there's a dominant, it's going to be in the front. We show dominant with an uppercase letter, and we show recessive with a lowercase letter. So we could have big T, big T, big T, little t. Notice that the dominant is always front here, or little t, little t. Now talking about probabilities. Probability is the likelihood of an event will occur. That's what we're going to talk about when we talk about crosses. The principle of all probability can actually be used to determine genetics and inheritance. Punnett squares can be used to predict and compare genetic variations that will result from the test. Homozygous versus heterozygous. Well, we know homo means the same. So it's organisms that have two identical alleles, or the same alleles, such as big T, big T. They're the same, big T, big T. Or little t, little t, little t, little t. They're the same, so they're going to be homozygous, or true breeding. Heterozygous, hetero means different. So this is going to have different traits, or different alleles. So big T, little t. These are also known as hybrids, because they're a combo of the two. A phenotype is the way the organism looks and behaves. It's what you see is the phenotype. Physical appearance is the phenotype. The genotype is the gene combinations. So the genetic makeup, the gene from the genotype. So phenotype physical, genotype gene. That's how you remember the two. Phenotype, you're going to say stuff like tall, short, green, round, physical appearances. For a genotype, you're going to give the alleles, the genetic makeup. Big T, big T! Little t, little t. Big T, little t. The genetic makeup, the alleles. So we're going to take a look at some white flowers versus some purple flowers on our pea plant. Purple is dominant, and white is recessive. Now, ladies and gentlemen, you've probably noticed that I have a little line above my P. Well, it's very hard to distinguish between a capital P and a lowercase p. So all of my lowercase letters will tend to have a line over them to distinguish that they are lowercase. These are the smaller ones. I suggest that you do that for my class so that we can keep it straight, and there's no ambiguity on whether that's supposed to be a capital letter or not. I can't really tell. If you put a line over it, I know it's lowercase. So, if I look at purple versus purple, well, purple, I've got two P's, and so when I draw my box, I'm going to put one of the alleles on one side here, and then the other one on the lower one. Now, this is one parent. This is going to be my first big P, big P. Now, the second parent goes on the top two boxes. One allele per box. So, my first big P comes here. My second big P comes here. And then we fill in the boxes by bringing the letters down and across. So I bring my big P down to here, and my big P down to here. And I bring my big P down to here, and my big P down to here. And then I bring it across. My big P comes over to fit here. My another big P comes over to fit on this box. My big P comes over, and my big P comes over. Now, inside of these boxes are going to be the offspring, or the probability of each type of offspring. So, this offspring is big P, big P. This offspring is big P, big P. This offspring is big P, big P. And this one over here is big P, big P. Now we know that the big P means purple. So all four of these are going to be purple. Now with white, again, all of mine have the little lines over them to show that they are lowercase. Again, I'm going to take my lowercase p and bring it over here, and my other lowercase p and bring it onto this side. And then my second parent, my lowercase p, comes here, and my lowercase p comes over here. And then I bring them down, and across, and across. Now, little p, well, those are both white and white, so it's going to be white. White and white, white and white, and white and white. So all four of these are going to be white. Now, let's look at heterozygous, where we see some hybrids. So we see a purple, but its genotype is big P, little p. So I'm going to bring my genotype over here and put a big P up top and a little p on the bottom. Now my other parent is also purple because it has a big P in it, but it's big P, little p. So big P goes up top here, little p comes over here, and then I still have to bring it down and across. So big P and big P. The only option of there is purple. Big P, little p. Now we have a dominant. We have a purple. So overall, this offspring is going to be purple. Because the dominant squishes the recessive and says, you are not going to show. Well, same with this guy. This one has a big P. It has a dominant, so it's going to be purple. But this one right here, little p, there's no dominance. So, the white is going to show. The recessive is going to show. Let's see if you can do one on your own. We're going to cross two heterozygous. Remember, hetero is going to be different. So when we're looking at the genotype, that means it's going to be a big T and a little t. Because that's different. And we're going to take the other parent, who's also heterozygous, big T, little t. Well, one of my parents comes over here, big T, little t. The other parent comes over and does big T, little t. Now pause and see if you can complete this cross for me. Go ahead. Pause. I know you want to. You do. And let's see if you got it right. So, what does this mean? Would this offspring be tall or short? Well, big T is dominant, and that's going to be dominant for tall. That one's going to be tall. How about these two right here? Big T is dominant. It squishes the recessive. So does the recessive show? No. So these two are also going to be tall. Then we have the point of the recessive. But they're happy because there's no dominant to squish them. So actually this offspring is going to be short. Well, that's it for now. See you later.